This is a picture of the beautiful blue Lake Erie. Wait, sorry, this is a picture of Lake Erie, and it is definitely not beautiful and blue. This green film is called cyanobacteria, or blue-green algae. It's currently wreaking havoc on thousands of freshwater ecosystems, including Lake Erie.
As a major tourist destination, Lake Erie can't afford to lose visitors to such an overwhelming problem. Nobody wants to visit a lake covered in a thick green sludge.
And that's not even the largest problem that cyanobacteria causes. They also cover the surface of water with so much algae that hardly any light is able to break through the surface. This leads to a chain reaction that can affect the entire ecosystem.
If light can't break through the surface of the water, then water-dwelling plants will not be able to photosynthesize.
These plants produce oxygen as a result of photosynthesis, allowing the water to be oxygenized so marine life can survive within it.
But with the film covering the water, the entire process is halted. Plants, animals, and other life forms lose their ability to survive in these so-called dead zones.
This chain reaction goes as far as to harm wildlife and humans, since ingestion, contact, and inhalation of cyanobacteria are also life-threatening and often fatal.
This can harm local industries because fishing and tourism can't take place on a lake filled with toxic algae.
Clearly, a solution to this problem is needed, but many of the measures taken to eradicate cyanobacteria, like chemical elimination, harm the environment just an equal amount, if not even more.
So, we have taken a different approach to the eradication of cyanobacteria, applying many biotechnological principles to do so.
And with that, we present to you our solution, the Het-enator.
This raft-style machine is designed to float across the surface of algae-infested waters, releasing electric shocks to induce a process called electroporation.
This electroporation induces cell competence, which basically creates holes in the membrane of a cell that allow them to take up foreign DNA from their environment. This is called transformation.
In short, transformation involves the alteration of a cell, in this case cyanobacteria, by taking up a small circular piece of DNA called a plasmid. This plasmid includes a gene that codes for a certain trait, basically changing the capabilities of what the cell can do.
In our case, we want to transform the cyanobacteria so it's unable to form heterocysts. This is the state of blue-green algae that allows it to fix nitrogen, or make it usable for the cell to use.
This is done in a process called nitrogen fixation, in which inorganic atmospheric nitrogen is transformed by heterocysts into organic nitrogen.
Bacteria require nitrogen to survive, so by taking away their ability to make safe-to-use nitrogen, they will die. But how exactly can we transform these cells to stop them from creating heterocysts?
To do this, we'll have to look at the individual proteins within blue-green algae. We'll be focusing on two, Het-N and Het-R.
Het-R is the main contributor to the production of heterocysts, while Het-N helps them to be maintained. But with too much Het-N, heterocyst production is actually stopped.
This means that the plasmid we insert into the cyanobacteria must increase the amount of Het-N present within it, or make the bacteria produce Het-N at a faster rate. If we do this, the bacteria will basically lose their ability to gain nutrients from nitrogen, and then they'll die.
With the plasmid created, you'll then have to actually add it to the bacteria. Transformations like this are normally done in the lab, but in this case, they'll be inefficient in killing the bacteria already existing in the water. That's where the Het-enator comes in. The machine works in a fairly simple basis. It starts by being introduced to the water and approaching a bloom. It then enters the bloom and sends out 8 kilovolt per centimeter shocks in 30-second intervals. 5 seconds after each shock, 50 milliliters of plasmid will be introduced to the water. This will be continued for a few hours until the bloom has been decided that it's been fully addressed. Then, the machine will either go back in to be restocked with plasmid, or it will move on to the next bloom.
The 5-second break between shocks and plasmid release gives the cells time to become competent, so they're ready to take up the DNA. From there, the overexpression of Het-N can occur, allowing Het-R to be overshadowed and heterocysts to stop developing.
And as we previously mentioned, without heterocysts, nitrogen fixation cannot occur, and the cells will not be able to have the nitrogen they need to survive. So with that, the blooms will die off, and with enough time, the problem will be pretty much eradicated.
Humans will have to help though. We plan to send out multiple machines to be piloted like a drone into the areas with the most bacteria. That way, all the blooms in the lake can be addressed, and not just one.
This method is better than most since it does very little to impact the ecosystem, as no chemicals or foreign organisms are being added to the environment. Plus, since their machines require human intervention, we have the ability to pull them out of the water at any point if something goes wrong.
But after many designs, redesigns, tests, and trips back to the drawing board, we've made rather certain that this technique will be incredibly efficient in the eradication of blue-green algae.
And with that cyanobacteria gone, Lake Erie and other bodies of water around the world could finally go back to being the beautiful blue bodies of water that they once were. And with that, my team would like to thank you very much for your time and consideration.